Okay, let's get into part 2. So in the first video, you would have learned about biotechnology tools, and in this video, we are looking at how we use those tools in biotechnology techniques. So there are heaps of techniques that biologists and geneticists use, and what we're going to be looking at are the ones listed here. So firstly, DNA recombinant bacteria. We did look at that in the first video as well. That was to do with plasmids. Importantly, we are going to be learning about polymerase chain reaction, also known as PCR. We're going to look at DNA sequencing, cloning, and DNA fingerprinting and DNA profiling. With some of these things were also mentioned in the other video. Okay, so firstly, recombinant DNA. So this process involves locating the gene of interest. So we use a probe for that. That's one of those tools. And using a specific restriction enzyme to isolate the gene. And then we cut the plasmid with the same restriction enzyme. That way the sticky ends are complementary. So we place that gene of interest into the plasmid, and then DNA ligase, remember that's the glue enzyme, will go along and bind those bases together, producing a recombinant DNA. And then we put this back into bacteria, and then the bacteria will multiply the gene, it will extract it, and use a... Sorry, that didn't make sense. And then we place that recombinant DNA back into bacteria to multiply the gene. And then, so we use a specific tool to insert the gene into a cell, such as a gene gun. So the bacteria might use that gene to synthesize proteins, or we might multiply the gene, extract it, and then use a gene gun to insert it into another cell. Here's just another diagram of that. So remember, using a restriction enzyme, we cut the gene of interest and the plasmid at the same location. And you can see here it's got the same DNA sequence. So AA, double TC, AA, double TC, right? Those sticky ends are complementary. We use DNA ligase, which combines them to make recombinant DNA. A similar process is agrobacterium technology. So agrobacterium is a bacteria that causes tumours in plants, but it's known for being able to swap its DNA with the DNA of the plant cell. So this has been used in the agriculture industry to basically infect plants with this bacteria that has a specific gene that's going to actually be advantageous for the plant. So very similar in the agrobacterium, we extract that plasmid, insert a gene of interest, so a DNA containing gene for a desired trait. So that might be resistance to pests, or being able to retain water for longer so that during drought the crops aren't going to die, things like that. So using restriction enzyme and DNA ligase, again, we make that recombinant plasmid, then we introduce it into plant cells in culture, and then we regenerate the plant, and that plant then has that new trait that we inserted into the plasmid. And the reason that that works is because remember, agrobacterium is known for swapping its DNA with the DNA of its host. So it swaps the DNA with the plant DNA. And so by doing that, then the plant contains that DNA, which has that gene for the desired trait, and it will then have that new trait. Okay, the next technique is polymerase chain reaction, or PCR. PCR is used to amplify DNA. So when we have a small amount of DNA, PCR allows us to copy it over and over and over again until we end up with probably billions of DNA strands, which are all the same, okay? So the materials that we need, or the ingredients that we need for PCR, firstly, we need the DNA that we're going to copy. That's the DNA template. Then we need DNA primers. These were mentioned in the previous video. Primers are small nucleic acids used to determine the start of the DNA segment to be copied. There needs to be free nucleotides in the solution ready to be chemically bonded together to make the DNA. We use a polymerase enzyme called taq-DNA, and this is from bacteria. And the reason we use this enzyme is because it can tolerate high temperatures. And we also need a buffer solution, which is just the chemical environment that this is happening within. So these are the 3 stages in the process. The first one is denaturing. So the solution is heated to 95 degrees to denature DNA into single strands. And I'm going to show that on this diagram. So we start with the DNA template, and then we heat up the DNA to about 95 degrees, which breaks the hydrogen bonds in between the nucleotides to separate the DNA strands. Okay. The next stage is annealing. So in the annealing stage, the solution is cooled to 60 degrees. Primers bind to single strands by complementary base pairing to provide a starting point. And the last stage is elongation. So the solution is heated back up to 70. We use that DNA polymerase enzyme that's heat resistant, and that is what attaches the free nucleotides that are floating in solution that are complementary to the exposed bases. So that's what's happening before we get to this point. The primers bind, that's annealing, and then elongation. The taq-DNA polymerase is attaching those complementary nucleotides out of solution. So if we start with 1 strand of DNA, we're going to get 2. And then in the second cycle, we've got 2 strands of DNA that are the exact same, and then we get 4. So after about 30 cycles, we actually get about 1 billion copies of this 1 double-stranded DNA. Of that original template, we end up with 1 billion copies of that. So advantages of using PCR is that it's much faster than using vectors to amplify DNA. So we might use bacteria to amplify DNA by inserting a gene of interest in a plasmid and putting that in bacteria, and then the bacteria multiplies, and by doing that we get the DNA will multiply. But PCR is much quicker. Only very small amounts of the target DNA are needed. Some disadvantages are that to produce a primer, we need to know the sequence of DNA segment of interest. So PCR is what is used when we get a COVID test, which is a mouth swab or a nasal swab, not the rapid antigen test. That's a different process. But when you get the swab, the scientists are using polymerase chain reaction to determine whether you have that virus or not. And what they're doing is they must have a primer for the viral DNA. So the DNA that's in coronavirus, they have a primer for that, and they're using PCR to multiply that, because if you only have a few copies of that DNA, it's going to be really hard to pick it up. But by doing 30 cycles of PCR, if there was 1 copy of that coronavirus DNA, there would now be 1 billion copies. And so that's much easier to detect, to determine whether you are positive for COVID or negative. A disadvantage of PCR is that it can only be used for short DNA fragments. Okay, the next technique is DNA sequencing. So this is a technique used to determine the actual sequence of DNA bases on the segment of DNA. And so this sequencing can be determined and compared with other species. So a lot of evolutionary biologists would use DNA sequencing to determine how species relate to each other. So the greater time that species have been separated from each other, the more time for mutations to occur and the greater difference in DNA. So if we look at this phylogenetic tree, the lion and the leopard are more closely related than the lion and the domestic cat, because every point is when there's been, this was one species and it's split. And then this was one species and that's split again. So there's been a mutation and natural selection has occurred at each point on the phylogenetic tree. So if we look at this, a lion has more similar DNA to a leopard than to a jaguar, tiger, or a domestic cat. And that's because the lion and the domestic cat have been separated for a much longer amount of time. And so a lot more mutations in their DNA could have occurred, making them more and more different. And we can see that in their DNA sequence. So their DNA sequence, the amount of difference tells us how long ago they were one species or how closely related they are. Okay, so another technique, we did look at gel electrophoresis yesterday. This one is a little bit different, and this is used to sequence DNA. So multiple amounts of DNA are placed in a beaker, and so all of these DNA code for a specific gene. And free nucleotides and modified labeled nucleotides are added. And so that's so that we can... So the different sized DNA fragments are injected into the capillary, so that's this tube. And the same as gel electrophoresis, which uses electricity, the shorter lengths of DNA will move through faster, and the modified labeled nucleotides are recognized by the detector. So that's how we can determine the sequence of the DNA, by using these modified labeled nucleotides. And just remembering that in all electrophoresis, the shorter strands move further along than the longer strands. Here's just another example of how DNA sequencing can be used to show the relationship between different species. So this table is showing us the amount of differences in the amino acid sequence of a protein called cytochrome. And so this is a protein that's in the respiration pathways of virtually all living organisms. So if we compare a human and monkey, there's only one difference in that amino acid sequence. If we look at human and turtle, there are 13 differences, and then human and yeast, there are 45 differences. So it's very clear to see that we are more closely related to a monkey than to rabbit, duck, turtle, or yeast, and we can do the same for the others as well. Okay, the next technique is electropherograms. So this is kind of a visual graphic way to sequence DNA. So you have to use a specific program, and if you do genetics at uni, I'm sure that you will do this at some point. So DNA sequencing enables mapping of species genomes. Okay, so electrophoresis is used to sequence the DNA, and then those results are displayed in this graph, which is an electropherogram. So we have these peaks which relate to a different nucleotide. What you need to be able to do is know that these results can be used to construct a DNA profile and be able to interpret electropherograms and tables of data that illustrate DNA profiles. So here are some examples. So this here, these are all the same gene, but you can see that visually we can straight away see that there are some differences, and so that's showing us a mutation, right? So each color relates to a specific nucleotide. In the next one, this one is different. So that base has changed, so that's a mutation, okay? And we can see that along here as well. This one here, this top sequence here, and when we compare that to this sequence down the bottom, we can see that there's a mutation there, so that G has changed to an A. And then same over here, we can see that that A has changed to a G. And so we can see that visually by comparing the graph as well as comparing the sequence. So electropherograms are used to identify mutations. That's one of their applications. They're also used to sequence DNA and to isolate DNA fragments and analyze those fragments. Here's just another example of the target DNA and then some other DNA samples so we can see where the differences are between the different types of DNA. And so this can also be used with different species of DNA. We can see where the differences are in their DNA sequence. There's some more information if you are interested in electropherograms. I invite you to pause the video now and go to this link. So on this link, this will go through gel electrophoresis, which is a technique you need to be aware of, and we did look at it briefly in the first video. So I'll just invite you to pause the video and go and have a play with that one as well. Okay, so the next technique is DNA fingerprinting. So in DNA fingerprinting, we're isolating DNA that is non-coding and very repetitive DNA. This is very unique to every individual person, and these sequences are called short tandem repeats, or STRs. Okay, so we use PCR to amplify these segments, and then we cut these DNA into fragments using a specific restriction enzyme, and then using electrophoresis, we can compare the lengths of these fragments, which makes like a DNA fingerprint or a DNA profile. So here is an example. So if we take DNA out of a plant cell, we extract it. It's missing a step here where we might amplify this DNA using PCR. We then cleave or cut this DNA using a restriction enzyme, and then we use electrophoresis. So maybe gel electrophoresis, which was discussed in the first video, to separate those DNA fragments based on size. Okay, and the following steps are just so that we can view this DNA fingerprints at the end. Okay, so if this was our sample and we're trying to determine whether this is plant A or plant B, we can very easily see that this is plant A. So if, for example, at a crime scene, if there was a plant that they need to identify, they might, or if there were just some fragments of plant, they might amplify that DNA using PCR, run electrophoresis so that they can then compare the DNA to other DNA of known plants to determine what the source of that plant is. Okay, so DNA profiling, very similar, is used for identifying criminals, custody disputes, fossil analysis, and genetic defects, and it uses the same techniques as DNA fingerprinting. You should know by now that smaller STR fragments, that's short tandem repeat fragments, move through the capillary electrophoresis tube faster. So remember, the smaller ones move faster, the longer ones are slower to move through the tube or through the gel. But the length of the DNA is small enough for PCR to amplify. So it's not a very long piece of DNA. So all humans have 99.9% of their DNA in common, and it is these regions called STRs where we have a lot of differences. There's a lot of variation. So that's why we use that for DNA fingerprinting. And by looking at the different positions on each chromosome, scientists can create a human barcode. Okay, so the DNA profile displays the genetic material at each loci, so each chromosome. Okay, and so law courts require 13 loci to be similar to, I suppose, determine whether someone is guilty or innocent based on their biological information. Here is a full DNA profile. So this is these are electropherograms of 4 different DNA samples, and this is just showing again so that this is what the electropherogram data might look like. Okay, so with these techniques, there are some ethical issues that we need to be aware of. Firstly, large companies might hold patents to control the knowledge and products. So people, if they hold the patents to control that like a technique or a product that's used for these techniques, then people might have to pay really expensive prices to access that technology. So if it's privatized and people can't access it, it's going to become really expensive. Individuals may manipulate their children to have more desirable characteristics, which is obviously an ethical issue. Animals may suffer when expressing the new gene. So if we use an animal as a vector, we might not know what side effects that's going to have for the animal. The organisms that are being the vector may have an allergic reaction as well. Transgenic products may have toxic effects that are as yet unknown. So if we put a gene of interest into a plant, we don't know that might have a toxic effect, like we don't know. Transgenic crops may be so successful they overtake native vegetation, and that reduces biodiversity, which is detrimental for our environment. It costs money to collect, establish, and maintain genetic information, and to undergo all of these processes is very expensive. And insurance companies or employers may be able to access the genome of an individual. And that opens up a lot of ethical issues. In particular, they might be able to deny employment based on your genetics or deny medical coverage based on your employment. Sorry, based on your genome. Okay, so next we have transferring genes between species. So transgenic organisms are when foreign DNA is incorporated into another organism. So a required gene is inserted into a fertilized ovum by microinjection, viral vector, electroporation, or a gene gun. So this is done so that the genetic information is modified so that an organism might acquire new or improved characteristics. Okay, so this process is called transgenesis, and it can be achieved chemically or physically or by using a virus or a plasmid from bacteria. Just in a bit more detail, microinjection is a fine glass needle injected into a fertilized egg. And a virus vector, what that involves is deleting the harmful part of the virus and inserting the gene of interest. The virus infects the cell with the DNA, and then it transfers the gene into the nucleus of the cell. And because it does this in a fertilized ovum, that's the first cell that's making up this organism. And then remember that DNA is copied into every cell that makes up that organism. So this DNA that's being injected will then be copied into every cell that makes up the organism. In plants, we spoke about Agrobacterium earlier, and so this is a naturally occurring bacteria that causes disease in plants, but what happens is the disease part is removed and the gene of interest is inserted, the bacterium infects the organism, and it injects the DNA into the plant chromosomes, and then the plant will express those genes. The other process is called electroporation. So an electrical pulse opens the pores. So you can kind of get that from the word electro and poration. Opens the pores of membranes and DNA enters into the cell. And the last one is a gene gun or a microparticle gun. So DNA is fired into the cell on an inert molecule, for example, gold, and the force of it forces it into the cell. Okay, gene cloning. So this produces large numbers of desirable genes, and what this involves is selecting and removing the desired gene, incorporating it into a plasmid from bacteria, and then as the bacteria replicates, that gene and the protein it synthesizes is also replicated. And as I mentioned in the first video, one commercial use of gene cloning is the mass production of insulin. Lastly, we're going to look at CRISPR. So CRISPR is a defense system that naturally occurs in bacteria. Bacteria uses this defense system against viruses. And so what it does is it's a system that destroys viral DNA that has been encountered before. So it recognizes and destroys the viral DNA that's been encountered before. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat, and it's being used as a editing tool for genes, and it has the capacity to edit parts of the gene by removing, adding, or altering sections of DNA, and it's currently the fastest technology and the best system for editing genes. When we look at chapter 1.8, we will look at CRISPR in a bit more detail. Okay, lastly, some applications of these techniques. So technology, this biotechnology is particularly used in agriculture, so by growing plants that have insect-resistant or salt-resistant plants, slower ripening of fruit, and also medicine, so the manufacture of human hormones in other organisms such as insulin, curing genetic disease, production of drugs.